All right, let's just jump right in. I want you to imagine a book. But this isn't just any book. We're talking about the book of life for every single living thing. This book is the complete instruction manual for building and operating, well, you. And it's written in a simple four-letter alphabet: A, T, C, and G. That book? That's your DNA. And let me tell you, this is no short story. We're talking about a text that's over 3 billion letters long. I mean, if you tried to print it out, you'd have a stack of paper 200 feet high. For decades, scientists knew this incredible code existed, but they had this one massive challenge. Exactly how in the world do you read a book that long, letter by letter, without getting totally lost? That's the big question we're going to tackle today, as we unpack the amazing technologies that finally let us read the book of life. So, our story kicks off with the very first breakthrough. This is the moment when reading even a tiny piece of DNA went from the stuff of science fiction to actual scientific fact. You can really break down the whole history of sequencing into a few major eras. We're going to start with that first generation, the one pioneered way back in 1977, and then we'll get into the next generation methods that honestly changed absolutely everything. So, the original method, dreamed up by Frederick Sanger, is just brilliantly clever. It's called chain termination sequencing. And the core idea is pretty wild. You don't read the original DNA directly. Instead, you build copies of it that get stopped or terminated at very specific points. So, what's the magic trick here? It's all about a special molecule called a dioxynucleotide, or DDNTP for short. A normal nucleotide, a DNTP, has this little chemical part, an OH group, that acts like a hook, letting the next letter connect. But the DDNTP is missing that hook. So once it gets added to a growing DNA chain, that's it. It's a dead end. The process just stops cold. It's the ultimate stop sign for DNA replication. Okay, so here's how it works. You take the DNA you want to read, and you split it into 4 separate test tubes. Each tube gets all the normal DNA building blocks, but you also add a small amount of one of those special terminator letters. So one tube gets terminator A, another gets T, and you get the idea. As the DNA copies are being made, they just randomly stop wherever that specific terminator gets added. Then, you take all those fragments you just made and run them on a gel, which is just a great way to sort them by size. And what you get is this beautiful ladder. The smallest piece is at the very bottom, the next smallest is just above it, and on and on. By looking at which lane each rung of the ladder is in, from bottom to top, you can literally read the DNA sequence, one letter at a time. It's just genius. Now, Sanger sequencing was a revolution for sure, but it had a huge problem. It was painstakingly slow and crazy expensive. It could only read one small piece of DNA at a time. And if you want to read the whole human genome, well, science had to figure out how to go from reading one page at a time to reading the entire library at once. I mean, just let that sink in for a second. The Human Genome Project, using this amazing tech, still took 13 years and cost almost $3 billion. This wasn't just a minor issue. It was a massive roadblock. To make genomics a real part of medicine, to study diseases like cancer, we desperately needed a radical change. And that change was next-generation sequencing, or NGS. And the difference is, it's just staggering. This was not a small step forward. It was a whole new ballgame. Instead of one DNA fragment at a time, NGS reads millions of fragments all at once. The cost and the time just plummeted, opening up brand new fields of science. The secret sauce, the big idea that made this huge leap possible, it comes down to 2 words: massive parallelism. Basically, doing millions and millions of tiny sequencing reactions all at the same time. Let's dig into how that's even possible. Okay, so first you've got to prepare the DNA. One of the common ways is called emulsion PCR. You start by chopping up the DNA into millions of tiny little pieces. Each one of those pieces gets stuck to a tiny bead. Then you mix these beads into an oil and water solution, which is a clever trick to trap each bead inside its own tiny water droplet. You're basically creating millions of microscopic test tubes, each holding just one bead with one piece of DNA. So inside each of these tiny little bubbles, that single DNA molecule gets copied over and over and over again. This is called clonal amplification, and it completely covers the bead with thousands of identical copies. Why does that matter? Because we've just turned a single, very faint signal into a really strong one that's easy to detect. All right, so now we've got millions of these beads, and each one is covered in copies of a unique piece of DNA. Now for the really cool part. How do we actually read the sequence on all of them at the same time? Let's check out two super clever methods. First up, we've got pyrosequencing. The pyro part means fire, and yeah, that's for a good reason. This method literally watches for a tiny flash of light every single time the correct DNA letter is added to the chain. Think of it like a tiny biological firefly. The chemistry here is just fascinating. So when the right DNA letter clicks into place, a molecule called pyrophosphate, or PPI, gets released. This PPI kicks off a whole chain reaction that involves luciferase, which, believe it or not, is the same enzyme that makes fireflies glow. The final result? A flash of light. The crucial part is, if there's no correct match, there's no PPI, and so no light. Simple as that. So a camera is recording these flashes from millions of beads all at once. The machine's output is called a pyrogram. A peak of light means you got a successful match. Now, this also shows one of its little quirks. If the next letters in the sequence are, say, GG, then two Gs get added, releasing double the PPI and creating a light pulse that's twice as high. Trying to accurately measure that height for really long repeats can be a bit of a challenge. Now, detecting flashes of light isn't the only way to pull this off. There's an equally brilliant method called ion torrent that uses a totally different idea. It doesn't need any cameras or glowing enzymes. Instead, it basically acts as the world's smallest pH meter. And the chemistry it's beautifully simple. When the DNA-building enzyme adds a correct nucleotide, it forms a new chemical bond. A natural byproduct of that reaction is the release of a single hydrogen ion, an H+. And what does a hydrogen ion do in a solution? It changes the pH. And this is the key innovation. The beads are put into millions of tiny little wells on a semiconductor chip. Right underneath each well is a super-sensitive ion sensor. So when a matching letter is added and that hydrogen ion is released, the sensor picks up that tiny change in pH as an electrical signal. It's an incredible way to turn chemistry directly into ones and zeros. So, from reading ladders on a gel to detecting tiny flashes of light and pH changes, wow, the journey of DNA sequencing has just been one of incredible ingenuity. But, no technology is ever perfect. These early NGS platforms were absolutely revolutionary, but they did have some trade-offs. For one, the length of DNA they could read in one go, their read length, was a lot shorter than good old Sanger sequencing. And like we saw with the pyrograms, they could sometimes struggle to accurately count long, repetitive strings of the exact same letter, like A, 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 A. But hey, those challenges just paved the way for the next wave of innovation. The power to read DNA so quickly and so cheaply has fundamentally changed biology and medicine as we know it. And it really leaves us with this one final, tantalizing question. Now that we can finally read the book of life so fluently, what profound new chapters are we about to write?